Hello everyone, I'm Belle, I'm the managing editor of MycoAsia channel. Today we have with us Dr. Arun Kumar Dutta. Arun is an associate editor of MycoAsia journal, and he is working as an assistant professor at Guwahati University, Assam, state of India. He has kindly agreed to give actually conduct an online, online workshop on free resources for mushroom taxonomy and phylogeny. Before I give a brief introduction about Arun, I would like to request the audience, the esteemed members in the audience, kindly follow the following instructions. I mean, you are already muted. You cannot switch on your video. But whenever we allow you to do that, please don't switch on the video, switch on the microphone, unless and until you are asking a question. Regarding the certificate of completion, at the end of the workshop, at the end of the workshop, we'll have an online assessment quiz. The link will be shared in the chat box. So please, be attentive, and you will have to score minimum 50% in the quiz to get the certificate of completion. If you miss the quiz, you are not going to get the certificate. So I'm making it very clear at the beginning itself. Okay, about Arun. Many of you may know Arun as a teacher, as a mentor, or as a friend. Let me give brief introduction about Arun. Arun did his bachelor's degree from University of Kalyani in the West Bengal. Then he did his master's and PhD degrees from University of Calcutta. And he is an meritorious. He has been a meritorious student and researcher. As you can see here, he has received DST Inspire Faculty Award. He has also received Young Scientist Award. Also received Prize of Excellence in Botany from the University of Kalyani. Also Reena Dutta Memorial Prize for highest marks in botany from the University of Kalyani. Before joining as an assistant professor at Guwahati University, he was working as DST Inspire Faculty Fellow at West Bengal State University. Arun's specializations are Arun's research interests are as follows: DNA barcoding and molecular phylogenetic analysis, mycology and plant pathology, and wild mushroom cultivation and prospecting. Presently, he is implementing a project on molecular systematics of wild edible macro-fungi of eastern India and the exploration of their nutritional prospects as nutraceuticals. Arun has already published around 60 plus papers, and many of the papers have been cited heavily. Our total citation is around 1,800, and he has an H index of 20. Recently, Arun and Professor Samantha, and along with some other researchers, they started a new journal called MycoKing, Journal of Modern Mycology. Arun is one of the managing editors of that journal. As I told, he's an associate editor of MycoAsia journal. He also serves as an associate editor of journal Species. Of course, he has been serving many journals as a reviewer, such as Phytotaxa, MycoAsia, Microtaxon, Green Studies in Fungi. With this introduction, I would like to request Dr. Arun Kumar Dutta to start his online training. Over to you, Arun.
Okay, thank you.
Let me stop presenting, Arun.
Yes. A very good afternoon to all of you, and special thanks to MycoAsia for giving me such a platform to connect with the mycology enthusiasts. And myself, Arun Kumar Dutta. Today, I'm going to present on the topic free resources for mushroom taxonomy and phylogeny. As the title implies, so we'll have some knowledge idea on mushroom taxonomy as well as phylogeny. Besides that, we will also have some knowledge on free resources. What are the resources from where we can have those things that will be suitable to aid in the identification of mushrooms as well as to perform the phylogenetic analysis. So before going to the details of the presentations, we should have a brief idea about the macrofungi. So what are these macrofungi? So but as the term implies, macro, that means the things that we can easily observe in our unaided eye. And according to Reed, the structure of a macrofungi should be equal or more than 1 centimeter in diameter. And these macrofungi are known by various names. They can be called sporocarps, basidiocarps, mushrooms. So here we can see different forms of macrofungi. First, we can see a clavaroid structures, or we may also find a coralloid structures. One of the classic example of these structures we can see in Clavaria as well as in Ramaria. So then this is a structure where we can see a clear lamella, and hence it's a lamellar macrofungi. So after that, we can also find out the tubular form of a macrofungi or woody form, or they may be cup-like, as we find out in many cases ascomycetes fungi. So they are called apothecial. Besides that, we may also find out some glabal structures. Here, this kind of structure we may easily encounter in the genus Astrias, where we can see there is an outer glaba which is completely breaks up, and this is known as the peridium. And besides that, we may also find out the stomatal structures as well. So if we consider a typical basidiomycetes fungi, so we can easily observe what are the structures there may be present. There may be a distinct cap, or we may call it a pilius as well. There is a long stipe region or the stem region. There is a vulva at the base, or a ring-like structures that is called the annulus. So here in the first picture, we can easily observe that this fungi is having a vulva, but there is no annulus. So the variation in the structures we may found in various kinds of macrofungi. Here only vulva is present, whereas in the next pictures we can see that there is a distinct vulva as well as there is a annulus as well. Whereas in the third picture there is no vulva, only the annulus portion is present. And there may be some fungi where both the things are absent. That means there might not be any annulus or any vulva. So if we now come to the mushroom taxonomy, so for proper taxonomical identifications, or we need several features. And if we categorize those features, so that may be morphological features or the molecular features. Now, within the morphological features, I have already discussed what the structures that we may encounter, and in microscopic structures, we may find out a forms of basidium if this is a basidiomycetes fungi. So beside basidium, we may also find out another structure that is known as the cystidia. If the cystidia is present in the pilia surface, that means the cap surface, so we will call that a piliocystidia. And if it is present in the lamellae, so there may be pleurocystidia or chilocystidia. So the cystidia which is present in the side of the lamellae is known as pleurocystidia, and the cystidia that is present at the end of the lamellae is the chilocystidia. So they may have different shapes, as well as we may also encounter cystidia in the stipe surface, and that is known as colocystidia. Besides that, if we look over the hyphae, so we may find out a clamp connection. That means it may be simple septate, or we may find out the clamp connections. So all these features are very much important. Now, here I have given a free resource. That means available things that to which you can compare the structures of your collected specimens. See here, there are lots of peculiar shapes. There are campanulate, conical, ambonate, or infundibuliform, or funnel shaped. There may be brain-like or umbilicate. So different forms of pilia structures we may encounter in various types of macrofungi. So if we have this chart and if we encounter any fungi, so we may easily say what is the pilia shape of my encounter fungi. See in the first picture here, this shape typically represents this one. That means here we can easily say that this fungi is having a campanulate pilia surface. Whereas in the next one, here we can say it is coralline structures, and in the third pictures, it is having a funnel-like appearance. So we will call it a infundibuliform pilia shape. So whenever we are going to the field, we should have to photograph well. Why? Because we know that there might be several developmental stages of a macrofungi. See here, this is one of the immature stage, and this is one of the mature stage. So the pilia shape varies from the immature one to the mature one. Here we can see that at the immature stage, the pilia is having a convex appearance, whereas in the mature stage, it is broadly convex or to somewhat aplanate in shape with a deeply depressed center. Now, besides this pilia shape, another important feature is the lamellae coloration and the attachment of lamellae as well. As the picture reveals, here we can see the coloration of the lamellae is to some extent brownish, and that turns into completely black or dark brown when it is mature. So, if we encounter only this specimen and lacks the remaining one, so we cannot easily say that what is the exact colorations of the lamellae. So, for proper identification, we need several developmental stages of a mushroom. Now here, if we see the picture of this is one of the specimen of Podaxis pistillaris. Here, this is the immature stage, and if we find out only this stage, it will be next to impossible to identify this fungus, as because it is immature one and also do not contain any spores. So in the developmental stage, this structure completely changes and creates a dome-like structure. Whereas in case of extreme maturity, we can find out a head full of spores, and this is known as the glabal part, where the outer layer is broken and the inner layer has been glabal part has been exposed. So for identification of these fungi, we need all these developmental stages. Now, if we concentrate over the pilia surface, what are the features of the pilia surface? Here is the reference structure that has been given by Lodge et al. 2004, and it is freely available in the internet. We can easily download it. So here there are lots of pilia surface characteristics has been mentioned. See here in the first this picture, here we can see some forms of squamules as it reveals here. So, and these squamules are tightly pressed with the pilia surface, and it is known as appressed squamulose. So, here we can see that there is lacks of squamous, and it somewhat looks like this one. So, striation. So, we may say that the pilia surface is striate in nature. And here we can see the pilia surface breaks off and it forms a areolations. That means we can have a pilia's areolation. We can easily compare it to the images that has been given by the Lodge et al. Now, this is the complete chart, and here is the resource from where you can get this chart. Here, lots of characteristics has been written down. So, a layman, and whenever they are collecting any macrofungus, so they can easily compare and say what are the shapes of the cilia, what are the shape of the gill, what is the attachment of the gill. So all the characters they may easily have for their collected specimens, and all these characters are very very fruitful for proper identification of a macrofungi. So if I photograph suddenly a macrofungi and if I send it identify this macrofungi, so it will be next to impossible to recognize or to identify the fungi. An immature stage, so that will not give a proper idea of the taxonomic identifications of that fungi. So we should have all the features with us and which will be handy for proper identifying a fungi. So here we can see, if we look over the gill attachment, here this is the lamellae. If the lamellae is style, so there may be various forms of attachment. See here, there may be admixed when the lamellae is just touched to the style. They may be adnate when the lamellae is broadly touched to the style. They may have decurrent attachment where the lamellae runs parallelly with the style, or even the lamellae may not touch the style, and in that case we'll call that the lamellae is free in attachment. So from this chart, what we have done, we have done a macroscopic character chart, and here all the features, that means date, collector, location, reference number, both subsystem, like this, all the features of a proper collection has been written down here. So here first we can see this is the growth substratum, which is very much important. I will show you in my next slides what are the growth substratum that we may encounter for a fungi. And then Pilias phase staining when exposed. So there are some certain fungi, whenever we pick those fungi, they immediately change their colors. And such changing of the colorations we can see in some species of Rusula. And for that feature, so in this feature is very much important. So what is the changing of the color when we just pick up the mushroom? And then there are also certain chemicals, the taste of those chemicals are very much important for their proper identification. So these are the macroscopic chart that we take with us whenever we go to any field, and all these features are to be mentioned for each of our collections. So from my view, collecting a huge number of samples is of less important than that of if you go and collect at least 5 or 10 samples, and if you have all these features for each of your collection, that will be much more easy or much more helpful for proper identifying of your collected specimens. Now coming to the different growth substratum, we can here this fungi grows on the wood. So here we'll call it a lignicolous. So obviously whenever we are identifying a mushroom, so first if we see that this mushroom grows on the wood, obviously we will not compare this fungi which grows on soil or any other substrates. So these are the important features that first we need to observe for our collected specimen. Here we can see that this is a species of Marasmias which have grown on the leaf. Here we can see there is a cow dung, and on which we can have some of the macrofungus. Here this is a species of Ascobolus. So obviously whenever we are collecting an Ascobolus from a cow dung, or if I am a layman and I am collecting a fungus from on the cow dung, so obviously I will not compare my collected specimen with those fungi that grows on wood or leaf. And here we can see that this fungi has grown on the soil and are known as humicolas. Here in the forest we can have we can observe that there are several termites. This termite nest harbors a special kind of mushrooms, and
Arun,
as the mushroom
Arun, sorry, Arun.
yes,
Have you stopped
hello,
sharing the slides?
no, it is
We can't see.
Okay, ladies and gentlemen, he is fixing it. He'll be back soon. Yeah.
So whenever we go into the forest, we might have some termite nest, and above the termite nest, we always encounter some of the fungi that grows on the termite nest. So here, as because this fungi grows on the termite nest, this fungi is known as by the name termitomyces. And here, if we dig up the fungi, we can have a long root-like structures. And this is known as the pseudorhiza, where at the apex of the root is closely attached to the termite nest. So if I am collecting a macrofungi that belongs to the genus Termitomyces, and if I do not dig up the fungi and do not show this long root-like portion, then it will be almost impossible to identify that Termitomyces species. So here, these pseudorhiza colorations might vary from white or brown or black. So it's an important feature to note down that what is the color essence of the pseudorhiza. Now, another important thing is the color changes on bruising. As we see here, these all three specimens belongs to the genus Agaricus. And the interesting feature is that for this specimen, whenever we bruise, we see a reddish coloration, somewhat reddish colorations of the stripe from the its white natural normal colorations. And here we can see that the color of the stripe surface changes to yellow. And in these specimens, we may not find any changes of the coloration. So this feature is also important. Here, if we go through the key that is available here in the link, so first characters for the taxonomic identification is the stripe yellowing distinctly when scratched. And then is contrast character is the stripe reddening, browning, or not changing when scratched. So if we do not have this data, whether what is the colorations of the stripe on bruising, we cannot move to the proper identification of the species of my collected specimen. Now here for this specimen, we can see that at the initial stage, the stripe was white in coloration, but on bruising it turns to somewhat bluish in color. So this is also an important feature to note down, and which will ultimately help for proper taxonomic identifications of this specimen. Besides these bruising characters, also there are certain chemicals with which the surface of pileus or stripe changes its color. Here all three belongs to the genus Agaricus, and here if we apply orthotoluidine, so we can see for these two specimens it turns to green in color. Whereas for this specimen, this turns to violet in colorations. So this is also a fruitful feature to note down, which will help for his proper identification. Besides that, we can also apply KH on the stripe surface or the pileus surface. We can easily see for this specimen, when we apply the KH on the stripe surface, it turns to yellowish in colorations. As well as for these specimens, whenever we apply KH on the pileus surface, it also turns to yellowish in appearance. So all these are the important ones that we will have to note down at the field for proper taxonomic identifications. Now, having all these characters with us, next thing what we will have to do, we will have to take the spore print. That means the colorations of the spores. So what for that thing, what we will have to do, we will have to cut the stripe and invert it on a plain paper. So if we do these things and if we keep it as it is for some certain period of time, so it will have its spore deposited on the paper, and we may have several colorations of spore print, and these color of this spore print are helpful for identifying your collected specimen up to the genus level as well as to determine its family. So once we have all these features with us, so we can fill up the field data sheet what we have taken for each of our collected specimens. Now, on completion of the macroscopic structures useful for mushroom taxonomy, if we look over the microscopic features, so we can see if we make a cross sections of the lamellae of a mushroom, so we can say several hyphal arrangement. Here we can see that the hyphae are irregular in arrangement and it is somewhat haphazardly arranged. So we call it an interwoven arrangement of lamellae or hyphae, trama hyphae. And here we can see there is a central parallel hyphae from where hyphae comes and joins. So it is somewhat convergent in nature. And here we can see that just reverse of this convergent. So it will be divergent, where the hyphae will run from the central hyphae towards its outer region. So arrangement of this hyphae is also very much fruitful for determining the family of the collected specimen. Now see, if we take the taxonomic key, so first point for determining the family is the hymenophoral trama bilateral. Bilateral, that means it may be these two types, either divergent or convergent, or the reverse character might be hymenophoral trama regular to irregular. So if it is bilateral, that means divergent or convergent, so we can easily see that along with that feature, if we take the spore pin colorations, we can easily identify the family of our collected specimen. So here, if the spore pin is white and the hymenophoral trama is divergent, so that will be under the family Ammonitaceae. So for a specimen which belongs to Ammonitaceae, obviously we will have this kind of hyphal arrangement in the lamellae trama. In contrast, if we find out this kind of lamellae trama and spore print is somewhat pinkish in colorations, so that will be categorized under the family Pluteaceae. And here we can see several specimens of Pluteus. Also, we can found this kind of hyphal arrangement in the lamellae trama in case of Valvariella as well. So after this lamellic trama features, another important feature is the basidiospore shape. So whenever we find out any spore, so now we will have to determine what is the shape of the basidiospores. And for determining that thing, there is a free resource that is available over the internet. So we can compare our observed basidiospores with this chart, and we can easily say what is the shape of the basidiospores of my collected specimens. Not only morphological visualization, there is also a chart that has been given by Largent et al. 1977, where they have determined the basidiospore shapes based on the Q value. So what is this Q value? Q value is nothing but the length divided by breadth of a individual spore. So for an example, if we have a length of a spore of 5 micrometer in diameter, and if we have the breadth same 5 micrometer in diameter, so what will be the Q value? Q value will be 5 by 5 equal to 1. So here, see here, Q value 1 to 1.05, that determines the globose shape. So statistically also, it will prove that you are having a specimen which have a globose basidiospore shape. Likewise, we may also encounter subglobose shape or broadly ellipsoid or ellipsoid shape, elongate and basidiform as well. Now, after determining the basidiospore shape, we will have to see what is the ornamentation of the basidiospores. And these also have a crucial role for identifying a species. See here, all these 4 spores belongs to the genus Rusula. And here, the first one is having a reticulate appearance of the basidiospore ornamentation. Whereas here we can see it is usually wing-like, where two wings are not interconnected. It is free. And whereas here it has wing type as well as these wings are interconnected to each other. So it will be wing little nets. And here it is somewhat dented in appearance and spiny. So we'll call it a varicose type. So all these four kinds of basidospores we may encounter in the genus Rusula, and where we obviously have to have this information, what is the ornamentation of the basidospores. Here we can see this one is a smooth wall ellipsoid basidospores. So here this kind of spores we may easily see in case of Agarica, several Agarica species. So all of them, they will have this ellipsoid smooth wall basidospores. And here we can see the spore is somewhat angular in appearance, and this is also an important taxonomic features with the help of which we can easily say to which family our collected specimen belongs. If we find out, for an example, if we find out an angular basidospores, we can easily characterize our specimen belonging to the family Enterolomataceae. Now, having all these morphological information that are that will help us for properly identifying a specimen, if we move over the phylogeny, so the basic things is for phylogeny is that we need to have a gene region. So what are the possible gene region that we may have for a mushroom sample? So here I've given some of the examples. So this one is the ITS region. Here this one is the 18S. So 18S region as well as the 28S region. This ITS region is in between two exon regions. That means the 18S RNA and 28S RNA. And this entire ITS region is also having an exon region that is known as 5.8S region. And these 5.8S region are very much genus specific. So if we amplify in 5.8S region from any of the mushroom, that will give a clear indication that to which genus our collected specimen belongs. Beside this 5.8S region, if we also amplify these two intron region, that means the intron 1 and intron 2 region, and according to the published paper of Scotch et al., these two intron region is having lots of differences between two species. So with the help of these two regions, we can easily discriminate between two closely related species. Other than this ITS region, we may also amplify this 18S region, which is also known as SSU region, that means small subunit region, or we may also amplify the 28S region, that is the LSU, large subunit region. Or there are some other protein regions as well, that those are RPB1, RPB2, etc. So whatever the gene of interest, we amplify for identifying our specimen. So after that things, what we need to prepare a phylogenetic tree to show the positions of our collected specimen. Now for this procedure, what are the steps? First, we'll have to extract the genomic DNA from our collected specimen. So we can extract the genomic DNA from the fresh sample, or as well, we can also extract the sample from the dried sample as well, specimen as well. So we can use the simple CTAB extraction procedure if we have the fresh samples with us, or we can use any of the available kits in the market. So once we extract the genomic DNA from our collected specimen, we'll have to now amplify our gene of interest from the genomic DNA of the specimen. Now here I have amplified an ITS region. So here we can distinctly see the bands of the amplified ITS region of my collected samples. So once we find out a distinct band, so now we can simply use the Sanger sequencing technology to sequence this gene region. And here we can see here, this is a simple chromatogram file that is showing the sequences of the amplified ITS gene region. So from this chromatogram file, we can easily have a length of sequence. Now, when we are observing the chromatogram file, we will have to see whether the peaks are sharp or not. Here, we can see different colorations. This is nothing but a simple Sanger sequencing procedure where they use different color dyes. And here, red is indicating the T base, whereas, the corresponding coloration that are showing, different bases color sequence. So, whenever we are observing the chromatogram file, we will have to see whether the chromatogram file is having a sharp peak or a biased peak like this. If we are having a sequence sample with us, and if we are having such kind of biased or hazy or overlapping peaks in the chromatogram, obviously we'll have to exclude and this sequence, as the sequence is very much of poor quality. So what are the software that is available for observing this chromatogram is the BioEdit. So there is a available link. We can easily download this BioEdit software, and we can easily observe the AB1, that is the chromatogram file of our sequence specimen. So once we get a pure or good quality sequence, so next thing what we will have to do, we will have to go to the NCBI GenBank nucleotide database, and a simple blast analysis will result to compare your collected specimen with all the available sequences that is deposited in the database, and it will give you a result, the base on which you can easily determine if your specimen has already been sequenced by any other person and has been deposited in the database or not. So once we find out this result, what we will have to do, we will have to now acquire the sequences of the close species from your specimen. So for this thing, what we will have to do, we'll have to simply open a notepad file, and in the notepad file, first what we will do, we will have to give a greater than sign, and this is a FASTA format. So you will have to arrange your sequence in such a format which will be useful for constructing the phylogenetic tree. Now first we will give a greater than sign, and then we will write down the name of the specimen. First we may write down the sequence specimen, and here in this format there is no space is allowed. So how will we indicate? We will have to use an underscore sign here. So this underscore will represent as a space, but if you space the give a space, so the software will not recognize and it will truncate the remaining things. So in the notepad, first we may write down the name of the specimen, then the GenBank number of that specimen, and then other information like the country and whether it is a type specimen or not. So like this way, we can prepare a data set, and such kind of data set will be later used for the alignment purpose. Now for alignment purpose, there are available free resources, and there are various softwares which we can easily use. Those are Clusterlex, Muscle, Map, and these are the available links from where we can easily download those sequences, those softwares, and perform the multiple sequence alignment. So once we perform the multiple sequence alignment, so we will have a such kind of file, and here as the sequence might be of different length, so we need to align the sequence, and upon alignment we can see easily that the bases are aligned in a systematic position. And if the sequence are of different length alignment, both ends we will have to trim, and we have to make the data set to a available length which will be covered by all the acquired specimen sequences. So trimming is also very much important. Otherwise, if you do not trim your alignment from the both ends, so what it will result upon alignment of the sequences. next stage, what we'll have to do, we'll have to find out a suitable model for my given alignment. So for this purpose, for finding out a suitable model, I hope you all know about the softwares that is available. We can easily download Mega, and Mega is a accumulations of various softwares integrated within it, and there is a special option that is the model selection. So where will we get that thing? We'll get that things here. So once we click on the model, such kind of option will come. That means find best DNA or partition model. So once we upload our data set here, and we click on this find DNA or protein models, so it will run a model test. That means the available model, it will run a test with the available models within this database, and will show you a result showing that what is the best appropriate model for your data set. And also it will give a statistical result, and in the statistical result we will find a BIC value. That means the Bayesian inference criterion value, and this BIC value indicates that which model is much more appropriate for your data set. Now, other than this Mega, another free resources is available over the internet, that is the Cypress Science Gateway. So here within this gateway, lots of softwares are there. We can easily use those things, and in the Cypress portal we may find out another software, that is the J Model Test 2, and we can also use this J Model Test 2 for testing the suitable model of our given data set. Now here, for the things, what we'll have to do, we'll have to upload the file in the Cypress Science Gateway, and which file? The alignment file. And if you upload your alignment file here in the Cypress Science Gateway, there is a required format, which is which will be read by this Cypress Science Gateway. And what is the format? That is the file format. That means the abbreviation of the file, that will be .phy, and this indicates that your file is in the file format. So we'll have to convert our data set to the file format, and we'll have to upload it here in the data section. So now next question arises, how we will convert our desired data set to the file format, right? So I have given a stepwise flow chart, which will speak that how we may convert our that data set to the desired or any of the format. Now if we have the text file, as we have downloaded the sequences and we have arranged in the notepad, what we will do, we can open it in Mega, or we can upload that text file to the Cypress portal. And from the Cypress portal, there is an option, that is the perform the alignment. So we can also do that alignment in the Cypress portal. There is also cluster available. Cluster is available there. Muscle is available, and there is also another software, that is the map. So we can also use map, muscle, or whatever you want. So with the help of that thing, you can have an alignment. So once you find out the alignment, you will have a FASTA format file. So that file abbreviation will be .FASTA. So now in the next stage, to convert the .FASTA file to the file format or Nexus format, what you will have to do, you will have to open this file with the help of AliView. So it is also a free software. We can easily download this from this given link, and this is a image that has been given here of the AliView. Once we open the AliView, and once we open our FASTA alignment in the AliView, next, if we want to save this file and then save as in various formats, save as Nexus, save as file, so we can easily save our data set to the any of the desired format. Okay, so once we save our data set to our desired format, Now we will have to perform the phylogenetic tree, and for that thing, what we will have to do? We will have to again select tool, and here we can see there are lots of tools available. As I was earlier mentioning, cluster W, there are various other softwares as well. Rally, all the softwares are available. Map, all softwares are available within the Cypress portal. So if we want to make the model test, so we will have to choose this one, J model test 2 on XCDE. So here, once we upload our file format in the Cypress portal, and if we click on this J model test, so it will ultimately find out the best model suitable for your given data set, and it will show you a result. Okay, so once we find out the appropriate model for our given data set, now what we'll have to do? We'll have to now perform the phylogenetic tree. And here in the Cypress portal as well, we can easily do the ML analysis. There are lots of analysis. I hope Dr. Raja has made you clear what are those MP analysis, ML analysis, or Bayesian analysis. So anyway, so if we want to perform maximum likelihood analysis, so what we will have to do? There is also a option, that is the RAXML HPC2 on XCD. So this RAXML, and it has been written down here, phylogenetic tree inference using maximum likelihood or rapid bootstrapping procedure. So once we click this option, so it will prepare a phylogenetic tree, based on the ML procedure, maximum likelihood method. And once it prepares the phylogenetic tree, what you will have to do? You will have to visualize the phylogenetic tree. And how you will visualize the phylogenetic tree? For this thing, you will have to download a software, that is the Fig tree. And here I have given a free resource from where you can easily download this Fig tree software. And if once you run this Fig tree software, if you open file and then open the tree that has been generated by that Cypress portal, so you can see a tree of appearance. So now what you can do, you can simply open this tree in your various formats, PowerPoint, or you can also can edit that tree in Adobe Illustrator. So with the help of various other softwares, you can edit or you can design your tree. You can demarcate the clades that has been generated, or where your desired species, just like this. I have done this for this specimen. What I have done, as this is a monophyletic clade, so I have just demarcated this clade to make it clear that my collected specimen belongs to this clade. And side by side, there are specimens from other genera where I have prepared some other colorations. So just like this kind of organization, you can suitably do in various available softwares. So once we perform all these steps, and once we taxonomically identify our specimen, and also we prepare the phylogenetic tree, the last one is very important, that is to deposit your specimen to a recognized herbarium. This is very much crucial. Whenever we go to the NCBI database on blast result, whenever we see the similar sequences, first our target remains that whether there is any voucher of that sequence or not. If there is no voucher, so there is no authentication of the specimen. That means someone cannot study that specimen. So that is very much important, that you need to deposit your collected or studied specimen to a recognized herbaria, so that it can be studied in the later days by any of the other scientists. So this is the design what we have developed for mushroom samples. What do? We take a simple plastic packets, and at the end of the plastic packet we keep silica gel, self-indicating course. We can see here the coloration of the self, silica gel is the blue, and this coloration remains until the silica gel absorb water. So once it absorb water, it will turn into white. And for mushroom specimen, preserving mushroom specimen is very tough, as mushroom specimen absorb water more rapidly. So if it absorbs the water from the surrounding atmosphere, what will happen? Other microfungi will infect your specimen and will make it rotten. So here we place the silica gel at the end of the plastic to give an indication whether the plastic is having any moisture or not. And we seal this by a sealer machine and keep a certain portion left as blank, and this portion will be in touch with that means the air can freely flow with the compartment where your specimen is placed. So here what we have done, we have simply covered our specimen and have kept over a tissue paper to make it more better representation. And then what we do, we completely seal the upper part of the plastic, and then all the details of that specimen we put down here, and then we finally seal the plastic at the upper of the your specimen tag. So this is a procedure, and this procedure keeps your specimen to be preserved in a herbarium in more well condition. And we have also published this procedure in in protocol exchange. We have given the detailed information how to preserve a mushroom sample and how to keep it in the herbarium for its longer use. So this is all about my presentations, and thank you to all for your patience listening.
Thank you very much, Dr. Arun. Thank you very much. It was a wonderful presentation. We will spend next 10 minutes probably answering the questions from the audience. Members of the audience, if you have any question, please raise your hand. We will let you ask your question. Yeah, please. Okay, first of all, Sheetal, do you have the microphone?
Yes, sir.
Yeah, please ask your question.
First of all, I would like to thank you, sir, for such a valuable presentation he has given. So my question is, you have said that we need to choose the model after alignment. So why is the need for selection of the model? I wanted to know.
So if there are lots of models available, and these models uses different algorithms to give you a phylogenetic tree. So which of the model is best? And whatever you do, there should be a statistical data that why you are choosing these models. So obviously, if you do not perform the model test, you will not have a statistically significant model which will support your alignment file. So if you have this model information and if you have the statistical information with you, you can easily say that why you are using this particular model among the available lots of models. If the algorithms are different, so it may be possible that the specimen, your concern specimen might come to a different cluster, or the support value might be of less. Okay, so it is very much important to find out the suitable model for the given data set.
Okay, thank you, sir. Thank you so much.
Ms. Ankita Rao, do you want to ask the question to Dr. Arun? Ms. Ankita Rao had a question. She typed in the chat box. Which primers we can choose for DNA sequencing for agaricales and polypore, Dr. Arun?
For agaricales and polypore, you can easily use ITS primer, and it gives better understanding and better distribution power. You can also beside ITS, you can also use LSU regions. So for this ITS region, what you will have to do, you will have to use ITS 1. And I am going to my slides. Just a minute. Here, if we observe this slide, I have written down the primer's name. Here, see, this is the ITS1 primer, which will bind at the end of the SSU region. And if we want to completely sequence this entire ITS region, so we'll have to use a primer which will bind at the end of the Eknes region, and here the reverse primer at the end of the 28S region. So if we use these combinations of primer pair, so these primer pair will help you to generate the entire ITS region that will comprise the ITS1, 5.8S as well as the ITS2 region. Now, if you want to amplify only this ITS1, that means intron 1 region, so for that thing, there is the available primer of White et al. That is you can use the ITS1, which will add at the end of the Eknes region, as well as there is another primer that is the named ITS2. So this ITS2 primer will bind at the starting position of the 5.8S region. So if you use these combinations of two primers, you can easily generate or amplify this ITS1 region. Side by side, if you want to generate sequences of ITS2 region, you can use ITS3 primer as well as ITS4 primer. Okay. And this ITS region helps well for identifying most of the Basidiomycetes fungi.
Thank you, Arun. Mr. Sridhar Pichar.
Good afternoon, sir. Sir, that the Indian agaricals, any new records, how to is find out, sir, that any checklist is available to in Indian agaricals overall in India?
So from Indian perspective, there is a book that is Fungi of India, although that is very much old, and although there is another thing, you can consult that mushrooms of India, fungi of India, from there you can find out, or you will have to find out the papers which is having a checklist in the checklist form. Likewise, if you try to if you are from the Kerala and if you find out any specimen, if you identify that specimen, and if you want to see whether that specimen earlier has been reported from Kerala or not, you can get the papers. A good work has been done by C.K. Pradeep sir as well as Munigon sir, and they have also published a checklist from which, with the help of which, you can easily see whether your specimen was earlier reported in the specific region. Those are the very much updated one. So if there is any updated literature available, there are lots of literature available. In the Northeast region, in the recent time, I have done a checklist of the available or earlier reported specimens. Although those checklists are solely based on the published information, but I have not got any voucher specimens. So here also comes a question: the authentication of the specimens. If there is no reference specimen, that means no one can study that. Whatever was the identification, you will have to rely on that. Okay. So you can go through your fungi of India as well as the latest available checklist of the region. Okay.
Okay. Thank you, sir.
Mr. Jackson, please.
Thank you, sir. I just want to ask, I work on wood decay fungi, and then I was fortunate enough to work on Heterobasidium perplexa, which was a rare species and found only in Nepal and India, and in India in Ceylon. While studying the morphological features, I find that Heterobasidium perplexa, when it grows on the dead wood, it grows only after like not even 1 meter or so, but very near to the ground. And then alongside, I work with Formitopsis, and that Formitopsis species, the height where it grows with respect to the ground is like barite. So can this criteria or can this characteristic be used as an identification?
See, you will first have to see the habitat. Okay, if the habitat is on the wood, whether that is close to soil, or that is much more above on the soil, and above part of a tree, this does not matter. You will have to see whether that is on the wood or on the soil. If it is on the soil, so obviously it will it will be categorized under humicolas fungi, and in that case you will not compare your specimen with those fungi that grows on wood. So, suppose for a group, if there are two specimens of a same genus, one grows on soil and another one grows on wood. If, for an example, so if you collect a specimen and if you see that your specimen grows on soil, so obviously you will go with the species that grows on the soil, and you will try to find out more similarity or other similarity with that specimen only. You will exclude that a species that grows on the wood. So that is also an important feature, obviously. And for proper taxonomy, it's a very complicated thing. Now, standardization of the characters is very much needed. So, for an example, if we see the Phileas cap shape, as I have discussed in my slide, so if we see the developmental stage, at the initial we can see a convex appearance of the Phileas surface, whereas in the maturity that turns to the infundibuliform or any broadly convex or plane. So for a same specimen we are getting different kinds of Phileas shape. So obviously this is not a strong character which will be much more useful for the taxonomy. So standardization of the characters are also very much fruitful or very much important for properly recognize a specimen.
Mr. Darshan, do you have the question? Do you want to ask the question?
Yes, sir. Sir, my question was that, sir, both of my species grows on dead standing tree, a gymnosperm, Pinus acacia. And then what I was saying that Heterobacterium perplexa, whenever I collect the sample, suppose for measurement, I took it with reference to the ground, it grows around like 15 to 20 centimeter above the ground on the wood while doing the measurement with respect to the ground. But for Formitopsis, whenever I found that it can grow anywhere, like it can grow near the ground on the wood itself, but then the height is very different. Sometimes it can have to grows along the standing dead tree high, but only for Heterobacterium, it grows nearer to the ground on the tree itself. So I was asking, like, can this be a characterization for the two species?
Yes, sounds great, Jackson. Yes, Arun, maybe you can have a personal interaction with him later. We have 5 more questions to answer, actually. Is that okay?
Okay, sir. Thank you.
Yeah, thank you. Now, Darshan, do you want to ask the question?
Yes, sir. Yes, sir. Good afternoon, sir. My question is regarding taxonomy at species level. Sir, is it possible to, by using only ITS, can we able to identify at species level?
See, initially when at the earlier time of the molecular phylogenic procedure, scientists used to use one gene region. And at the starting of the Scotch et al. Paper, when they showed that ITS is most fruitful for proper identifying, properly discriminating between two species. And if you go through that paper, you can see that they have sample across a wide range of fungal families, and I have find out why this region is much more fruitful for properly discriminating between two specimens. Other than this ITS, now scientists are more prone to use multiple gene regions. Other than this ITS, there might be, you can use LSU region, you can use SSU region. Although LSU and SSU region is the species discrimination power is comparatively lesser than that of the ITS region, and also you can go for protein coding region. that is RPB1, RPB2, or you can also use TF1 alpha transcription elongation factor. So at the basic level, ITS is enough for differentiating between two species. And if you are not getting more differences based on the ITS sequence of your two of your collected specimen, then also only you will have to think over other regions, which will help you to properly discriminate between those two collections.
Yes, sir. Thank you, sir. Another, sir, when you go to the field before taxonomy and before characterizations, so what is the short-term strategy to preserve the samples to bring from field to lab, sir?
For that purpose, what we do, we usually use a 4-square box, and we put those specimens within that 4-square box, and we just close the box lid. And otherwise, you will have to take keep in mind that your specimen do not comes to the open air. Otherwise, what will happen? Your material will be rotten. So for that thing, what you can do, you can simply put your specimen in a plastic. You can use you can tightly seal the opening surface opening area of the plastic. And you will have to keep in mind that that specimen do not get in contact with the open air only. Okay.
With the plastic, sir, we had faced the problems of degradation within a short term, sir. In many cases, we are facing degradation.
So no, you can use reusable plastic, no problem. Suppose you are taking that thing, with you, you are again cleaning, and again you are going to the field with that same plastic. Or you can side by side you can use 4-square box, which is also reusable.
Yes, sir. Yes, sir. You are right regarding this. Thank you, sir, for the question. Answer.
I request the audience to restrict to only one question, because we need to take 5 more questions at least.
Okay, sir. Okay, sir. Thank you, sir.
Thank you. Next, session, Ambikesh.
Thank you, sir. Good afternoon, sir. Sir, my question is, can we use this molecular technique for identification of micro-fungi?
Yes, obviously. No, why not? You can use these molecular techniques for identification of microfungi. There are lots of papers available. And also suppose if you have a culture of a microfungi, you can extract taxonomic DNA from that microfungi, and with with using of simple setup procedures, and you can go for the amplifications of the your desired gene regions. No problem. You can apply molecular techniques for the micro.
And sir, there is another one. Just asking that, can we use this technique for identification of lichens? Because in lichens, the majority part is fungus. So can we use this for the molecular identification of lichens or not?
See, there are lots of papers. Also, in the plant groups, they are using this ITS region, for identifying these plants. There are also, papers with the help of that ITS primer, ITS region. They are, identifying the bryophytes and other, groups as well. So obviously, you can use this ITS like that.
Mr. Sazina, please.
Hello, sir. Thank you so much for this wonderful presentation. Sir, my question is, actually, when I was in my graduation studies, I worked on mushrooms like Basidiomycota, Garicale members. And suddenly I went for an interview, and there was a panel sitting asked me, while you was collecting your mushrooms, can you tell are these poisonous or not? And then I forget about this question. Now my question is, sir, when we are visualizing mushrooms in field, so could we determine either they are poisonous or not?
So determining the mushrooms at the field level is next to impossible, whether that is the poisonous or the edible. So, for my presentation, I think it is clear what are the things, or what are the characters, bunch of characters that we need for properly identifying a specimen. And once you get to know what that specimen is, unless and until you cannot say that that is a poisonous or edible one. But as we know, most species of Volvariella are edible. That means all species of Volvariella are edible. Whereas in case of Amanita, most species are poisonous. Very there are some species which are very least amount of species which are edible. So if you find out any Amanita specimen, so it will be next to impossible for you to say whether that is a edible one or poisonous. Or if you find out a species of Volvariella, you can guess that Volvariella, that genus is not having any species which is poisonous, so I can eat, at least I will not die. Okay, and at the field, it is impossible to recognize whether that one is a poisonous one or the edible one. Yes.
Okay, sir. Thank you.
We will take up last two questions. Mr. Asit Mahato, please.
Good afternoon, sir. I have a question, sir. When I study anatomy of some Marasmias species, sir, I not found any spore in microscope, sir. Or when I found some spore that is not perfectly visualized, sir. Why, sir? Kindly tell me, sir.
See, what are the difficulties that occurs in case of Marasmia species? That is very much small, in structures. And another thing is that, compared to any other, macro-fungi.
Bluetooth mode.
Hello. Okay. So in case of Marasmia samples, that specimen gets dry so easily, and they do not need any extra care to make them dry. And what's happened when you are drying? If the sample is much more older in your, if you keep the dry sample for much more longer times, what will happen? The spores that gets fallen from the basidiocarp, from the lamellae. So that that is the reason why you get a little amount of spores in your sample. So I will recommend here to study the specimen once you come from the field immediately. Okay, so that will help you to get the spores of for that your collected species of Marasmia.
Mr. Sushant Bharna, please. Last question.
Yeah, good afternoon, sir. Am I audible?
Yes, yes, sir.
Yeah, sir, for color differentiation of any mushroom, there is a book called Methuen Handbook of Color, but that is not available. So is there any other substitute for this book, or we can get this book anywhere from anywhere?
Yes, there are free available of the color book. So I will share the link. Although that will not be comparable with that of the Methuen Handbook of Color. That is the standardized book, and I know the prices of that book is very much. So no one, very few of us can afford. So I will share some of the color links from where you can download those things. Okay.
Thank you so much.
Thank you, Dr. Arun. And thank you, audience, for all the questions. I know you may have a few more questions. Arun will answer them at the end of the practical session. Arun, maybe we'll take a break for 5 minutes and we'll come back in 5 minutes. After 5 minutes.
Okay. See.
Ladies and gentlemen, we are taking a break of 5 minutes. We'll be back back by 13:22 hours Indian Standard Time. Thank you.
Okay. So am I audible?
Yes, you are.
Okay. Fine. So now, once we have a chromatogram file, I hope my screen is presentable to all of you. This is a chromatogram file, and the indication is that there is a .ab1 which has been written, and here if you have that bioedit installed in your computer, and if you have such kind of a AB1 file, what you will have to do, you will have to double click on it to visualize the code. So I have already double clicked it, and let's see what is the result that is coming. Now here we can see a chromatogram file here, and here we can also observe that the peaks are sharp, okay, and clear enough, which indicates that the sequence quality is good. Okay. So what we will have to do, we will have to take the sequence from this chromatogram file, and we will have to exclude the biased region here. So for that thing, if you look at the starting region, you see the peaks are not continuous and are not sharp enough at the starting region. Okay, so for that thing, what we will have to do, we will have to exclude those biased regions. So if we see here, from here we can get a sharp peak. Okay, so for this thing, what we will do, we will just select the sequence from here, and like this way I have excluded the remaining part, and I am just selecting the sharp peak sequences from the chromatogram file. So once we go to the and select the chromatogram file sequences, what we will have to do, we will have to simply copy it. And we know if we press Ctrl C, that means it will copy the sequences. Okay, so like this way I have already copied the sequence from here in the chromatogram file. So next, what will I have to do, I will have to open a notepad file. Okay, so like this way I will open a notepad file, and as I have told in my presentation, first I will have to give a greater than sign, and then the name of my specimen. Suppose I will write down here specimen 1. Okay, or I can give specimen underscore then 1, and then I can write down the voucher code of my specimen, or the collection number. Suppose collection number AKD 110. So this is the information I have given for my specimen. Now next I will give an enter, and I will paste it. Simple Ctrl V will paste your copy sequence from the AB1 chromatogram file to the notepad. Okay, so once I have pasted the desired sequence bases into the notepad file, the next stage what I will have to do, I will have to see what are the closer species available in the database that are similar to my specimen. Now I will move to the NCBI database. Okay, so for that thing I will write down NCBI in the address bar, and just like this kind of page will appear. So here see here there is an option blast, and we will have to click on this blast. So once we have clicked on this blast, so here there are lots of options: nucleotide blast, protein blast. So obviously we have generated the nucleotide sequence. That means our sequencing gene region is a nucleotide. So obviously we'll click on the nucleotide blast. Now once you click on the nucleotide blast, just like this kind of page will appear, and here at the this place, entire query sequence. Query, that means your sequence, specimen sequence. So here we'll have to paste our sequence. So we'll simply use Ctrl V again here. So like this way we have pasted our amplified sequences of my specimen here, and in the next what we'll have to do, we will have to hit the blast option. So for this thing we can directly click on the blast option, or if we want to keep this page and want to open the blast result in a separate page, what we will have to do, we will have to click on show result in a different window, and once we have clicked it, now we will have to click on the blast option. So see here a new page has appeared, which will show you the possible similarity of your specimen sequence with the all available sequences in the database. Okay, now it is searching over the database. And see here, it has given a result. Okay. So we can see here the first one of my species is Volvarella sp. There is a voucher number AKD 2022. Okay. Then GB GUBH 19922. So like this way, we will find out the similarities of the available specimens sequences in the GenBank database with our sequence. So here, what we will have to do, we will have to see the percentage identity, this column. Okay. So here we can see that this specimen matches with my sequence with 100% identity. This 100% denotes that your specimen is nothing but this specimen. That means this one and your specimen is similar. If there is any name for this specimen, so obviously this name could be applied for your collected specimen. But here we'll have to see the authentication of the specimen. So once we click on this sequence, so what we will find? We will find an alignment file. Okay. So let's see the alignment file. It has been given a alignment identities as well as gaps. That means here, if we look over the identity, see out of 655 nucleotide phase, it has matched with 100%. That means there is a full match of your sequence with the deposited sequence, and there is no gap. That means all the bases is available for your sequence and the sequence that was earlier deposited in the data. And here for this specimen, we have got the detailed information from this heading. Okay, what is the name or what is the voucher number of this specimen? Once we click on this sequence ID, so it will again open a new page. Okay, so in this page we can get more detail of the deposited specimen. Okay, so once we click on here, we can see these who are the persons who have done this who have deposited this sequence, and is there any has the paper has been published in any journal. That means if the paper has been published, so we can recommend it as an authenticated source. But here also we'll have to see whether suppose while we are dealing with the taxonomical identification, if we see paper has been published in other journal, a nutrition journal, so obviously the taxonomy is not the first criteria, and possibly might be the reviewer who has reviewed that paper might not be the expert of the taxonomy. So there might be some cases of the misidentification. So here, for choosing the sequences in the gene base, we'll have to be very cautious whether the sequence is an authenticated one or not. Suppose if I have observing a sequence of the Rusula, and if I see that Professor Bartbach has deposited that sequence, and that person is one of the renowned person on the taxonomy of Rusula, so I can believe on the sequence that obviously that is authenticated identification, as because that has been done by a well-renowned person in that genus. So other than this thing, publication information, we will also have other information like country, okay, the exact location, the date of deposition of the specimen, okay, as well as who collected this specimen. So here, what we will do, we will simply copy this name. So Volvarella sp AKD 2022. So we have copied it from here, and we will go to our notepad file, and again we will give a greater than sign, and then we will paste this one. Okay, and see here between Volvarella and sp there is a gap. So here, as I have told in my presentation, you will have to give a underscore other than this space, as because the software will not recognize the space. So again here, so like this way, we'll give space, underscore. So after this thing, we can add the voucher specimen number. So here, what is the name voucher specimen number? GUBH 19922. So we can again copy it, and we can provide this information here. Okay. So again, an underscore. And then another important thing is the region of origin of this sequence. And here we can get the country of the deposited sequence. So here we'll copy this Indian. Okay. So once we have copied all these informations, another information we'll have to copy, that is the what is the accession number for this sequence. So again we'll give a underscore sign and we'll collect the accession number. See here, for this sequence, another column is there, that is gen bank, then one number, and then 0.1. So we'll exclude this 0.1. So what this 0.1 denotes? This 0.1 denotes that this is the deposition which has not been altered by the author. See here, suppose you have wrongly deposited a sequence against a name, and later if you find out that you have wrongly deposited the sequence against that specimen name, so you can request GenBank to change the sequence. Okay, so if that kind of cases arise, so here what you will see the accession number and then 0.2, not 0.1. So 0.1 is signifying that this sequence has been deposited by the author and has not been changed or altered in the later time. So if you change the sequence in thrice, so there will be 0.3. Okay, so once you get the accession number, you will just copy the GenBank accession number for this sequence. So again I am copying it and I am pasting it at the end of this specimen, and then again and enter. Now here I will have to paste the sequence of this specimen. See here, it is in the different format, but see here my specimen has having another format. So what for this thing, what we'll have to do, we'll have to click on the faster. See here, there is an option faster. So once we click on the faster, what we'll observe? We'll observe a sequence here in our required format. So we'll again then copy this one and we will paste it here. Okay, so like this way we can have as much as sequences we want to acquire from the GenBank database. Okay, suppose if you're other than for molecular phylogeny, other than the similar sequences from your database, while you are performing the molecular phylogeny, you have a particular aim. You should be a particular aim. That means suppose your specimen belongs to a different section or belongs to a section or subgenus of a mushroom genus. So for that thing, what you will do, other than the similar species, you can go through the protologs, you can go through the books, and you can see what are the other specimens that is earlier taxonomically characterized belonging to their section or subsection or their subgenus. So you can manually search in the internet and you can manually download those sequences. So for that thing, what you will have to do, you will have to simply go through the here and see here nucleotide. Suppose if I want to download another sequence, suppose Volvariella bombicina, okay, and want to add that sequence in my data set. So what I will have to manually entry Volvariella or bombicina or Volvacia. Okay. So once you write down the name, so this will show you a lots of sequences of Volvariella volvacia, and see here there are lots of other gene regions available as well, but you will have to be cautious that what is your amplifier gene region. So as in my case the amplifier gene region is the ITS region, so here we'll have to manually choose only the ITS gene region of Volvariella volvacia. Okay, so here for sorting the sequences, what we can do, as we have already come to know that Volvariella ITS region is having 5.8s region in between, so we can filter the searches. Otherwise, see here page 1 to 83. That means page 1 to 83 of the result, is having the sequence of Volvarella volvaceae of different genes. So here, what we'll do, we'll add, other than this Volvarella volvaceae, as we know, 5.8s region is in between intron 1 and intron 2. So what we can do, we can add 5.8s. Okay. So, writing down these, if we, press the search button, see here, now the sequences will come, which is having the ITS. As because, see here, 18S ribosomal RNA gene, partial sequence, then ITS 1, then 5.8S, then ITS 2, and then 28S region. See here, all the sequences has come, which is only having the ITS region. Okay, so here in this case, if we want to acquire other sequences from the GenBank database, what we will do? Now we will click on here. See, Volvarella volvaceae voucher. There is a specific voucher number. That means this specimen has been deposited in any of the herbarium. So we will have to choose this one. But see here, Volvarella volvaceae strain this. So we can also manually check whether this specimen has been deposited or not, or someone erroneously identified the specimen and just deposited the specimen against any of the name. See, GenBank do not filter any of the sequences. So whatever sequence you will deposit in the GenBank database after the name, this is your responsibility, to make it authenticated, so that the others can blindly use your sequence. So here, if we click here or Volvarella volvaceae voucher this, we can get detailed information here. So this specimen has been deposited from Thailand, and it has been done by Kumla J and Nokorin. So this Nokorin is one of the good worker when the taxonomy of the Volvarella is concerned. So we can blindly believe on this sequence. So what we can do, we can likewise again we can copy this Volvarella volvaceae and paste in our notepad file, and we can add the voucher number here after the name, and then the GenBank number, and then clicking on the FASTA file, we can have the sequence, and also we can also have the information from the country where it has been deposited. So like this way, we can prepare a data set. Now, if my specimen belongs to a specific section or subject, so taxonomically, based on the morphological feature, we have identified the specimen belonging to that section. But here in the phylogeny also, you will have to prove. That means you will have to show others that yes, your specimen belongs to that section. Okay, not also to the other section. So how will you perform this step? Now we will go through the books. Suppose a genus is having 5 sections. Okay, so what you will do, you will take the similar species where your specimen falls along with the representative members of the other sections in your data set. And what is the purpose? The purpose is nothing but to show that your specimen do not cluster with the members of the other section. And like this way, you can easily visualize, you can easily present that what is the systematic position of your specimen. Okay, so like this way, I have already prepared a data set based on the search result. So this was my species Volvarella bilobata that has been recently published in the journal Phytotaxa. So these 2 are my sequence, and then based on the blast search, I have taken the sequences of Volvarella, as well as I have gone through the books, and have seen that what are the close species or what are the morphologically look-alike species. Might be 2 specimens might be morphologically similar, but they are genetically dissimilar. So if they are genetically dissimilar and distantly related, so obviously on blast search they will not come together, or the blast result will not show you that that species. So in that case, what you will have to do, you will have to go to the NCBI database and we'll have to manually search. As because a layman, whenever they found any species, first they go through the morphological features and then only the molecular feature. So if two specimens are morphologically look alike, so we will have to add that the sequence of that species. Okay, so like this way we have added the specimen sequences of several species of Volvariella, and other than this genus, if there is any closely related genus, suppose earlier all these specimens were characterized under the genus Volvariella, but in the recent past, there is a scientist who has break down some species of Volvariella and have raised a new genus, that is the Volvoplutea. So obviously Volvariella is to some extent related to that of the Volvoplutea, but there is some difference. So what in that case, what we will do, we can also have some representative members of the genus Volvoplutea as well. Okay, and once we take a few representative members of the closely related genus, we will have to take a outgroup taxa. Now see, outgroup taxa, taking the sequence of the outgroup taxa is very important. Why this is very much important? Because for an example, if in a class there are 5 standard students and 7 standard students, both are mixed together and sitting in a room. Okay, so if a layman come enter to the room, he cannot easily distinguish that which students belong to class 5 and which students belong to class 7. So but if a teacher enters to the room, he can easily segregate the standard 5 and 6 standard students. Okay, so here we will have to take a somewhat related species. So for this outgroup taxa, what is the purpose of this outgroup taxa? This will show you the placement of the in-group taxa. And in this case, what are the in-group taxa? As your specimen belongs to the Volvariella, so obviously the remaining members of the Volvariella are the in-group. Okay, and other than that thing, which will help you to segregate the species, all acquired species of the Volvariella into a cluster together, that is the outgroup species. So here we have gone through the earlier, if there is any earlier published literature available, so you can choose the outgroup taxa from that published literature, and you can mention in your written paper that we have chosen this specimen as the outgroup taxa based on the study of this disease. Or if there is no earlier paper, so in that case what you can do, there is a mother paper, that is the major clades in agriculture. So you can go to the that paper and you can see a huge phylogenetic tree. So you will have to search that in which clade your specimen belongs, and you can take easily next to your clade if there is any closely related taxa genus. So you can get the information from that phylogenetic tree that has been presented by Moncalvo et al. And that is also easily available over the internet. You can freely download and you can see that which genus is close to your collected specimen genus. Okay, so like this way we have here for Volvariella specimen, we have chosen Cantharocybe species as the outgroup taxa. So like this way we have prepared a data set. See here we have prepared the data set in the notepad format. Okay, so I hope you all have learned the mega in your last presentation. So first today I am focusing on the that Raxema Cypress portal. Okay, so we'll do the after this arrangement of the data set, we will have to do the alignment. Okay, and after that alignment we will have to build tree. So directly we will go to the Cypress portal. I have already given the link of the Cypress portal in my slide. So or you can manually type over the internet and you will get here a Cypress portal. Okay, so once you click on the Cypress portal, this kind of page will appear, and see here use the Cypress Science Gateway. And once you click on this use the Cypress Science Gateway, it will lead to a different page here. Okay, so if you do not have a user ID or password, you can easily register here and can have a user ID and password here. So I have already registered and I have a user ID and password. So I will log in here. Okay. So I have here a login here. So next stage, what I will do, I will now upload my notepad sequence here. Okay. So for that thing, what I can do, see here, there are lots of things. So it will not be mixed. So first one, what I will have to do, I will have to do a create new folder. So here I am creating a new folder, and suppose here I am naming the file like demonstration. Demonstration. Okay. So I am now saving this file. And see, once we have saved this file, so look at the left column. Under the demonstration, there are two parts, data and task. Now, if we click on the data, then see there is no data. Okay. And likewise, if you click on the task, you can see there is no task. So now what we will have to do, this is our data that we have already prepared in the notepad. Now we'll have to upload that data here. Okay, so we'll go to data section and we'll click on upload or enter data. So this kind of page will appear, and here we'll have to choose file. So for demonstration, see a ready sequence. I have named that ready sequence notepad file as the ready sequence. So I am now saving here. So I have opened it, and now simply you can click on save button. So see here that file has been uploaded, and you can see here the label ready sequence.txt. That means the text file. Okay. So now once you have uploaded your notepad file here with the sequences, now you'll have to go to the task button. And here you will have to create a new task. And what is the purpose? Our purpose is to make the alignment. Okay. So now here we'll give a description, that is the alignment. Okay. Alignment. So once we have given a description, now we will have to input a data. Now we'll have to click on select input data, and once we click on this select input data, we will see that file that you have uploaded. Okay. And see here we have used that greater than sign, all these things. And what is the format? It is telling that it is in the FASTA format. Okay. So what we will have to click on here, and we will have to select this data. Now select data. So after that selection of data, now this option will come. This is the select tool, and here we'll have to see or the software that we will use for multiple sequence alignment. So here there is mapped. Okay, so we can use this map for the alignment. So we simply will click on mapped. Now we have chosen the map tool for alignment of our data set. Now here see input parameters. There are lots of parameters. That means they are saying that there are 51 parameters set. So now you cannot save and run task unless and until you go and manually visualize the parameters. Okay. So for that thing, we will have to click on this parameter set and see what are the parameters. There are lots of parameters for the data set, and here we will have to change certain things. So what are the changes we will have to do? Here there is maximum hours to run. Always make this thing as 0.50. Okay. So in default there is 0.25. That means how many times it will consume to generate the result. This is fits well 0.50. So now we are using the MAP version 7.4.90 and then output format. How want your file in the output format. You can click on, if you want cluster W format, you can click here, or you can, if you want FASTA format, so you can click here. So I need my alignment file to be in the FASTA format, so I have clicked here. Now see here, input type, that means input data type. So you can click here, determine automatically. That means the software will determine your data, whether that is the nucleotide sequence or the amino acid sequence. Okay, so you can click on the determine automatically, or you can manually click the nucleotide if you are having the nucleotide sequence. So here I am giving the determine automatically. Okay, and remaining things we keep as unchanged. Okay, and then we will go to the output option. See here, output order, same as input or align. Now what is your purpose? Your purpose is to get an align. So you will have to choose, click here, align. If you keep same as input, so there will be no alignment, and it will be as it is as you uploaded. So here you will have to click on the align. Output guide tree. Do you need tree? No, not needed. So now here we will save the parameters. Okay. So once we have saved the parameters, our required parameters, see here, lots of parameters has been reduced, as because there was lots of other parameters. What are they? Protein model. If your sequence is a protein, so amino acid, there are lots of options are there. So that option will be omitted now from the total number of parameters. So it will be obviously reduced for your nucleotide sequence. Now, once you have selected the input parameters, you will have to click on this button, save and run task. So I am clicking here, save and run task. Now see here, alignment file, parameters we have selected, and action view status. So here, if we click on view status, we can see that our file has been added to run in Quake, and then command rendered successfully. That means your file format is fine. That's why it has been stacked to the software, and then it is successfully input file stacked successfully. Okay, so we have successfully uploaded and our file format is okay. Now we will come to the return to task list. Once the task is completed, this option will be changed to view output. Okay, and to see this view output, we will have to refresh the task in the meanwhile. So we will have to click this option, refresh task. See here, once we have refreshed the task, this action option has been changed to view output. Now, if you click on here, view output, you will have a data file. Okay, and here there are lots of information available. And here, which file you need? You need this output file. See here, output file. So this file you will have to download. So we will download the output file, and this file has been aligned using that of the muscle, that of the MAP software. Okay, so we will manually go to the file and we will copy that file, and again we will put that thing to our folder. Okay. Now we have copied that output file here in the our phylogeny folder. Now see here, what is the extension of this file? Output.mafft. Okay, and this MAFFT is not a readable format for the alignment to be understood by the software, available software. So what this case, what we will do, we will make a copy of this file and here. Okay, so we have copied here, output, copy, and then mark. So we are keeping this as a data file. Now simply we will have to change the extension. So what we will do, we will have to rename the file. What we will do, we will keep the output as it is, and here we will have to delete this mark. Okay, so we'll have to change this extension, and here we'll have to write down pasta. Hope I am clear. So I am here changing the file extension from .maft to .fasta, pasta. Okay, so once I have changed it, so there will dialog box will come. Do you want to change it? Yes. So I have changed it. See here, once I have changed it, now I can see here. It has been changed to pasta format. So if I want to visualize now, so simply if I have the softwares already installed in my computer, I will have to double click on it. And this alignment will be opened with the softwares. Okay. So here for the softwares, I have already Ali View, as I have already discussed in my presentation slide. I have Ali View installed in my computer. So I have opened the alignment file with the help of Ali View, and see here all the species names has been appeared and their corresponding sequences. Okay, and the sequences has been aligned here. Okay, so if we go at the end position and the starting position of the alignment, we can see here this specimen is having a long sequence. Okay, whereas for the remaining specimen, the sequence is of less in number. So in that case, we can easily see that the sequence is of different length. So for this thing, what we will have to do, we will have to delete some of the position. Okay, or even if we see that some of the sequences are quality of the some of the sequences are less, so we can also delete that thing from here. So for an example, suppose if you see here, we can see lots of starting sequence is missing. Okay, and this sequence is starting from here. So our next target is to make all the alignment sequences to a equal length. Okay, so for that things we will have to do trimming of the sequences. So here we can select here, and then we'll suppose select the remaining portion and we will delete it from the alignment. Now for that thing, what we will do, we will select on edit option, and then see here there is option delete selected. So once we click on delete selected, see all the starting unequal sequences has been deleted, and here what all the sequences has been started from a particular point. Okay, so likewise we will go at the end position, and here we will have to see maximum position. What is the position where most of the sequence bases are present? If we see, then this is here at this point. So what we will do, we will now select the remaining part. Okay, and again we will click on that edit and then delete selected. So this portion has been deleted, and now all are coming with same length. Okay, so once we have done this thing, now what we will have to do, we will have to save this file in various formats. Now see here why saving the file in various formats is required, because there are several softwares which recognize a particular file format. So this is very much crucial. So here we will get the option. If we want to save this alignment as FASTA, we can click here. If we want to save this alignment as Nexus file, we can click here. If we want to save this alignment as file, we can click here. So for the Cypress portal that recognize the file format, so obviously I will need a alignment file that is in the file format. So I will click on this file. So once I have clicked on file, see here the abbreviation output.phy, and this phy indicates that the file will be in the file format. Okay, so I am simply saving the file, and the file has been saved in that your folder. Okay, if I want to save it in the Nexus format, so I will have to click on the Nexus and see here the output.nex. The extension is the .nex. This is the Nexus format, and this Nexus format is required for the softwares like Mr. Base, and also this Nexus format is also required for the softwares like POP. Okay, so if you want to perform phylogenetic work in POP or Mr. Base, so you will have to save this file, alignment file, into Nexus format. Okay, so because that those softwares will not recognize your .py format, that means the file format, or any other format. So I am also saving my alignment in the Nexus format as well. Okay, so here I have already saved our alignment file into output.nex, that means the Nexus format, and output.py, that is the file format. Okay, so we can also see whether it has been saved. So remember one thing, any file format, you can easily open any file format in Notepad because that is the default one. Okay, so if you want to see the alignment, so what you will do, you will click on right button, and then you can see what is there in the file format and what is the difference between file format and Nexus format. So let's see, open with Notepad. Notepad is a thing which opens any of the file format. So if you open to Notepad, see here, this is the file format where first there is a number, that is the number of taxa in your data set, and then there is a number, that is the number of bases or alignment length. So what is the total number of alignment length here? 976. And what is the total number of taxa in your data set? That is the 46. And see here, all the files, alignment things has been saved here. Okay, and at the last, there is all the your specimen sequences, and at the starting there is these two information. See here, file format. If any software is recognizing the file format other than this information, this will not recognize the file, because that software needs how many number of taxa is available in your data set and how many number of characters are available in your data set. So unless and until these two things are available, so those softwares will not recognize your data set. Likewise, now we see what is the Nexus file format looks like. Again, open with Notepad. See here, Nexus, in the Nexus file format, starting there will be Nexus writing, and then there will be certain codes. Okay, and these are begin data, then dimension, then end text. The softwares we recognize the Nexus format. If you write down 46, it will not understand. There needs this syntax, end text. Only then those software will be able to recognize that the number of taxa in your data set is 46. Okay, and if there is a abbreviation written in chart, that means number of characters equal to, then that this will be the number of the characters, and then data type DNA and gaps, that means the this symbol indicates the gaps, that means these are the gaps. Okay, and if there is any missing data, that will be shown as the question mark. Okay, so in the Nexus format also there are certain things that are needed at the end of this thing. So you need to convert your data set to the desirable format, which will be read by the various software. Okay, and this Nexus file is needed for, as I have told, for POP software, for Mr. Base. Okay, and but that XML, that means the Cypress portal need not not the next format. Cypress portal need the file format. So now what we will do, we will again go to the Cypress portal, And now in the data, we will now add that data in the file format. So now again, we are uploading the align file. Again, upload data, choose file. And here, which one? That dot file. Okay. Then we are opening, and now we are saving. So our that file has successfully been uploaded in the portal, in the data folder. Now we'll move to the task folder. Okay. And what is our aim? Our aim is now to find out the model. Okay. What is the best model for our data? So for that thing, what we will do again, we will create new task, and then we will write down suppose model test. Okay. So now select input data, and here you will have to select this file, not this .txt file. Okay, that txt file, we use this txt file for the alignment purpose. Now we have got the alignment file, and we need to select this file format. Okay, now select data, and then we'll go for the model test. See here, there is J model test 2 on XDE. So we'll click on here. Okay. So once we click on the J model test, here also lots of parameters will come. So we'll have to manually see the parameters. So we'll click on the parameters. And again, if we click on the advanced parameters, long parameters will come. And here we'll have to do certain changes. So maximum hours to run here, 0.5. Okay, fine. So specify the name of your output file, output.txt. Fine. Now here, clustering search, BIC, all these will keep as default. Okay. What are the things we'll change? Base tree topology, we'll have to select it maximum likelihood. If there is default, by default there is non-mandatory selected, so we'll have to make it maximum likelihood. And then information criteria, select information criteria. That means which information criteria you need. That means which statistical information you need. So for this thing, I need Bayesian inference criteria. You can also select AIC value, no problem. So if you select this Bayesian information criteria, so a lots of model will come, and it will show the Bayesian information, data for the model. Now we have selected it, and what we will do, we will simply click on save parameter. Now we have saved the parameters, and now see, there is ultimately 18 parameters that has been set by. Now I will click on again save and run task. So once I click on this software will perform the model test, and I can check the status. And if I see success, my data has been successfully submitted, so my file format is okay. Okay. Or if you see that if there is no success, so it will be, if you do not upload the file in the required format, you will not get this message. The input file stacked successfully. Okay. So now we will come to return task list, and we will wait until again we see here view output. So in the meantime, we will refresh the task. So it will take some time, and the time taken depending upon the number of specimens in your data set. If you have lots of specimens in your data set, specimen sequence in your data set, so it will take much more time. And if there are few sequences, so it will take much lesser number of times. Okay. So let's wait. Up to this, is it clear to all? Yes, sir. Okay. Similarly, the time that is been taken by this, portal, we may have a, some knowledge on mega. If someone have missed last day of Dr. Raja's class, okay, so we can also perform this model test. We can also perform the alignment and build tree using the mega. Then that is most easy one to use that of mega. So for using mega, what we will do? We earlier we'll have to install mega in our computer, and okay, as because it is taking time, so I am going to the mega. Okay, so once we have opened this mega, now we'll have to click on align. Okay, so now again we will click edit or build sequence, build alignment. Now see a dialog box have been arised, where we'll get create a new alignment, open a saved alignment session, retrieve sequences from a file. Okay, so if you want to create a new alignment, so you can click on open a click alignment. Once you click on create a new alignment, you will get a dialog box containing that this one. What is your data? That is whether that data is DNA type, protein, or any other. Okay, so if this is DNA, so again another dialog box will appear, and here what we will have to do, we will have to click on edit option and then insert sequences from the file. So once we click on insert sequences from the file, what will happen? We will have to go to that folder, seminar, then for demonstration, and then that ready sequence.txt. Okay, so you can visualize the available files here. If you can't see the notepad file here, what you will have to do, you will have to click here. Okay, and here are lots of things. If you want to see visualize the FASTA file that is available in your folder, you will have to click on FASTA. So see, once I have clicked here FASTA, that output.FASTA file has been appeared here. So if I want to do alignment, so we click on text, the notepad file that we have first prepared. So we'll click on notepad and then open. So once you do, see here all the things has been opened here. And here interesting thing, if you look over the sequences, so here you can see the bases are haphazardly arranged. See here ATGC, it is a haphazardly arranged. Okay, so and also some of the specimens is having a long stretch, whereas some of the specimen sequence size is much more less. Okay, so now here if you save this file and if you run the phylogenetic tree, obviously it will not give a good phylogeny. Why? Because the bases are not arranged systematically. For arranging the bases systematically, here is the necessity of the alignment. Okay, so here we'll click on the alignment. If there are options, two options here in the mega, you can use cluster W, you can use muscle. So muscle is much more, the algorithm used by the muscle is much more better than that of the typical cluster W. So for this thing we'll use muscle. So it will select here and a dialog box will come. So we can use here the default parameters that is available here. So simply we'll click on compute. So once we click on the compute, it will align your sequence here and it will give you a result. Okay. So we'll come later on and we'll see the result here. So now again we are going to Cypress portal to check whether that task has been completed or not. Okay, fine. The task has been completed in Cypress portal. Okay. So here we'll click on view output. Now once we click on view output, we'll see lots of files here. And we'll have to see that what is the required file. That means your required file here is the output.txt. That means the output file is your required file. Okay. So here you can download the file or you can view the output file here. Okay. So if you want to view, you will have to click on view, as because it is in the text format. Now see, you will see here the things with lots of information. The output file with lots of information. And for the information, number of sequences is there, number of nucleotide sites is there, 976. And see here how many models available. There are lots of models available. And there are combinations, several combinations as well. See here, TRN plus G. G, that means the gamma. There are several names. You can get the abbreviation. What are these models' names are there. So these lots of models has been tested with regard to your alignment file, and you will get a data based on the statistical analysis. So you will have to go at the end. Now see here, model selected. It will show you model selected. Which model? TRN plus I plus G. Okay. So this model has been selected by the J model test as the best model for your data set. Okay. So here, see why this model has been selected. Here are the names of the models, and see here there is a column, BIC column, and here are some data. Okay, and see the best model is the model which has the lowest BIC value. This value of this model is the lowest one, then that remaining, and it is chronologically arranged. At last model, which is that there is a high BIC value. So obviously this is not the statistically significant model for your data set. Okay, and here is the necessity why you need to test the model. Otherwise, what will happen? You can use any other model, and you can erroneously do start doing the phylogeny. Now here TRN plus I plus G. So while we are writing down, then why we have chosen this model? So we can write down. We have selected the model TRN plus I plus G for the given data set based on the lowest BIC value of this one. And this information will give a clear indication of the purpose why you have chosen this model. Okay. Beside that thing, you will also get other information for this model. See here, if you want to run this model in the POP, so information set of information has been given. So first you will have to write down begin POP, then LSET. These are the codes, and base, these are the base, NST equal to 6, and so you can simply copy it and you can paste in the POP folder. If you are performing the phylogeny helping using the POP file, okay, or if you are using the other model, no problem, you can use information. See here, NST equal to 6. So this NST equal to 6 in base, Mr. Base also, you will have to write down this manual NST equal to 6. Okay, then only the software will understand that which model you are referring. So now we have come to know what is the best model we have got here. Okay. So we can apply this model for the data set in any of the available softwares for preparing the phylogenetic tree. We can do that task here, or we can do the task here in the Mega. So for that thing, what we'll have to see here, this work has also been completed, and see alignment has systematically arranged your sequences, and you are having a star mark at the above portion of the nucleotide bases, and this star indicates at these positions all the sequences is having T base. Okay, see, all the sequences having T base, And this alignment is also very much important to understand the deletion and insertion regions. Likewise, for example, suppose if you see for this sequence, for this specimen, Volvilla bilobata, this one, this sequence is available, whereas for the next one, Volvilla pleotica, there is a huge gap. That means we can interpret that either this portion has been deleted, or in the reverse, we may also think that this region sequence has been inserted for this specimen. Okay, so this alignment will let you know to understand that what are the deletion, duplication, or insertion sites in your data set. Now here also you have got a long stretch, so you can also trim your sequences here. So you can manually select like this way the end portion, and you can simply use the cut, that means Control X option. So once you click on press the Control X, it will cut the remaining end. Okay. Likewise, you can also do that thing from the starting portion. If you want to cut this section, you will again use Control X, so it will cut. So likewise, you can trim the sequences. Anyway, now my target is to do the phylogeny, as we have got the model, specific model. The model test work you can also do here. Model, find base DNA protein model. You can find the model here. Now we have already found a suitable model, and what is the model? That model is TRN plus I plus G. Okay. So now we can build the phylogenetic tree using this thing, but before that thing we'll have to upload that thing here. Okay, but these mega format understand only the .meg file. So for that thing, you will have to save your file in the .meg format. So for saving the file in .meg format, what we will have to do? We will open a file or session. We have saved that FASTA file. We can open analyze the nucleotide sequence. Okay. Protein nucleotide sequence? No. So I have opened the file here. Now I can see the alignment. See here. You can visualize that aligned file. Okay. Export data. Okay, to convert the file into MAG format, you will have to perform the alignment here in the mega file. Otherwise, you cannot convert the file into the .meg format. Okay. So here we had the sequence, aligned sequence, but we have missed to save it. Again, you will have to do that thing. After file, we have got this from the Taxonomic software, Cypress portal. Now we will have to cut both the ends as we have done. We can simply use Control X from both ends to trim both sides. And here we can save this data. See here, save session, export session. Under the export session, here you are getting the mega format, FASTA format, POP format. Okay, so here only you can save the file in mega format. See here, .meg format. Okay, so we are naming mega format the file as a mega format, and we are saving it. Input file data, no, not needed. Protein coding region, no. So we have already set the alignment file in the mega.meg format, and see here, this is the file that we have saved. Okay, and for performing the phylogenetic work in the mega file, you will have to open that make file. Okay. So now we'll have to open a file, and which file will open? That make file .mg mega format file. So we are opening that here. So the file has been opened here successfully. We can see this is the file that has been opened here. Okay. So once the file has been opened, we have got the suitable model, that is the TRN plus I plus G model for the maximum likelihood analysis. So what we will do, we'll click on phylogeny and we will do construct or text maximum likelihood tree. So we are clicking on this thing, and here a dialog box will appear which will have number of options. Now here some of the things you need to change. So among these options, none or bootstrap, you will have to set the bootstrap method. That means how many times you want to create the tree. Okay. So in bootstrap method, suppose if you click here 100, so this software will prepare the phylogenetic tree 100 times. Or you can manually give 500, 1000, that depends upon you. For a demonstration purpose, I am giving as much less as possible. So I am giving here 50. And now see here model or method. Now I will have to click here. And what the model? TRN plus I plus Z. This was the best model. And this TRN, this means Tamura Nei model. Okay. And I, that means irreversible, and G, that means gamma. You can get all these things in mega. The details, of these abbreviations. What is the name? Detailed name of the model. So we have got TRN, that is Tamura Nei plus I plus G. So we will set this option here. So see here, there are lots of models: Juke Cantor, Kimura 2 parameter, Tamura model, Tamura Nei model, general GTR model. So which model we got? Tamura Nei model. Okay. So we have selected this thing, and then we have got I plus G. So here there is another next column, rates among sites. So we will click on here, and here you can see the option only the gamma distributed has invariant site or gamma distributed with invariant site. So we found the best model as the G plus I. So we are clicking on G plus I. See here. So we have successfully set the desired model TRN plus G plus I as the result was shown by the J model test. Okay. So once we have selected all these things, what we will do? We'll click on compute. So it will generate a phylogenetic tree. Okay. So this mega will now generate the phylogenetic tree using the specified model that you have found based on your mixture model, your J model test. Let's wait. Or if you want to perform the model, phylogenetic tree in the Cypress portal, similarly you will have to upload that file and you will have to run the tree. So again, I will go to the task for obtaining the ML tree. Now we have already installed uploaded the alignment file. So we'll do the create task. Now here you can do ML3. You can write out ML3. That means we want to create ML3. Now select input data. We had already earlier, uploaded the file. So again, we will select on that data. Select data. So we are performing the J model test on the file as well as we are constructing the phylogenetic tree on the file as well. Now, if you want to create the phylogenetic tree here in the software using the RaxML software, so what you will have to do, you will have to search. Here is the option RaxML HPC2 on XED. So you will have to click on here RaxML. It is taking time. Let's see. Is the phylogenetic tree has been completed in your mega? And see here, using the model, a phylogenetic tree has been prepared using the mega software. And here, I hope you can remember, we had used a outgroup taxa in the data set. But see, in mega, there was no option to mention what was the outgroup taxa in our data set. Okay, and that is the major drawback of mega. We can, although we can do it like this, Canthrocybe, this was the outgroup taxa in our data set, and we can place it, place root on bunch, but these do not seem to be the perfect way of doing the things to make it out from your phylogenetic tree. Okay, but in RaxML there is an option where you can determine the outgroup taxa. See here, now we have chosen RaxML HPC2 in on XCD. Now we'll click on the 50 parameter sets. Now click on the parameter set and we'll again change the parameters as we record. The website is down. Okay, so we have got here. So here select set parameters. We'll set some parameters. Maximum hours to run. I have earlier told we'll change this to 0.50. Now enter the number of patterns in your data set. That means you will have to write down here how many number of patterns is available in your data set. So now what is the number of patterns? If you open your file with the help of notepad, you can easily see what is the number of patterns. That means the number of characters 976. So we'll copy it, okay, and we'll provide this information here in the number of patterns in the data set. So we have set here 976. Okay, and then please select the data type. Your data type is the nucleotide, not the protein. So we'll change it to nucleotide. Okay, and see here there is an option outgroup. See here in the RaxML you can define the outgroup. Okay, so here we will have to mention who is the outgroup in your data set. Now we will go to the align file and we'll see what was the name of the outgroup taxa. Canthrocybe. Okay, now how it appears. Canthrocybe. this one, Canthocyte. So we can copy the name from here of the outgroup taxon. Otherwise, what will happen if we write down Canthocyte name and miss any of the symbols here? So the software will not be able to recognize the sequence about which you are talking about. Okay, so I am just copying the name as it is written in the alignment file, so that the software can easily understand about which taxon I am mentioning as the output. So I have copied it, and simply what I will do, I will paste it here. So see here, it has been pasted. The details name of the output taxon has been pasted here. Okay. Now once this has been pasted, so next what we will do, we will now select on some other parameters like this. Choose model for bootstrapping. You can say use the default option GTR cat, or you can use the GTR gamma. Now then you can have the bootstrapping option, number of bootstrap interaction. You can do it 1000, 100, that depends upon you. And preferably 1000 bootstrap support is preferable. So you can make it 1000, and after giving all the parameters, you can now save the parameter. Okay, so now we are saving the parameters. We have already set the output taxa, and now we are save and run the task. So the file has been, we'll check whether the file has been successfully submitted. Yes, it is successfully submitted. So the software will perform the phylogenetic tree and will give you the result. Okay. Now here in the Mega, we already got the phylogenetic tree. Okay, but here the main drawback is that we did not mention what is the output taxa at the starting of the phylogeny. That's why what happened, that taxon came within the within your input taxa. If this is a closed species, this problem occurs. But if the output is so far distant from all the in-groups, so obviously it will come outside, and that does not signify that that is facing a proper thing. Okay, if both the genus are very much close, so obviously there might be this kind of problems where output taxa will be placed within the in-group. Okay. So from Mega, we can safely save the image as the PNG file or the PDF file. So you can save this tree in your required format. Preferably you can save it as the PDF file, so that it will have the higher resolution of it. Okay. And also, if you want to get some information on this tree, there is an option. See here, caption. There is a caption. So if you click on this caption, it will show you the detailed things that you set. Okay. This is a Tamuranai model. That means the tree has been prepared using Tamuranai model, and where you have given GTR, I. Okay, all this information will be shown here. So you can simply copy it and can use it in your manuscript. Okay. Anyway, so this is the way how we prepare the phylogeny with the help of Mega. Okay. So now if we move to that of XML, so let's check whether the tree has been prepared or not. Yes, it has been prepared. The tree has been prepared here. So now we click on view output, and see here lots of files are here, and here we will have to download this file, XML bipartitions.result. Okay, so we'll manually download this file, XML bipartitions.result. So we have downloaded this thing. Now we'll copy it and we'll paste to our folder. So this is the tree file. Now see here, you cannot see this tree file. You double click on it. It will ask you which is the app to use it. Okay. So for observing the tree file, there is another software, that is the fig tree, that I have already discussed in my slides. But and also I have given the link, source link, from where you can freely download the fig tree. So now I have already installed that fig tree in my computer. Now I will open that fig tree. Fig tree. So I have opened. Now I am opening the fig tree. This is the fig tree file. So once you open it, double click on it, will open. And with the help of this fig tree, you can see the tree that has been prepared by the taxonomy. Now for that thing, what you will have to do, you will have to file, open. You will have to go that folder where you have saved your tree file. So this one, rational bipartisan result. Now you will click on open. So once you click on open, it will ask you the node or branch level. Okay, how do you want to name the node or branch level? You can write down bootstrap or label, whatever you want. So we are writing down BS. Okay, now if we open, the software has opened a phylogenetic tree. I hope you can see, all of you can see. You can zoom it. There is an option to zoom. You can see. Okay, you can manually edit here as well. You can make the taxon names in your desired format. That means you can change the font size, font, everything. So if I want to do suppose Arial font, so I'll click on Arial and then 16 size. See here, the font has been changed. And you can also expand a tree so that two specimen will be there will be ample of gap from one another. So now we have got a tree, and see here in this tree there is no support value. Okay, so at the starting there is an option how you want to show your name your the support value. So we named it as BS. So where we will get it? We'll get it at the node levels. See, this is the node level. Okay, we'll have to tick it, node level. And if we click on, or if we expand the node level, there will come node edges. So whatever the value is showing, this is the node edges. Okay, then node height, you can show this thing, or you can also show the bootstrap support that you name. So I have named the bootstrap support as BS. So once I click on BS, see here the bootstrap support value has come in. Okay, so for individual clade, we now have got the bootstrap support value. So once we have done the editing here, so what we will do, we will now save this file as in the PDF. So we will now convert this file, export PDF. Now we are saving it, ML3. We have saved this tree in our folder. Now we will go to the folder ML3. So we have saved this file in PDF format. If you have PDF installed in your computer, so if you double click on this ML3, you can choose Acrobat and it will open. See, the tree has been opened. And this tree has been prepared using the XML in the portal Cypress. Okay, and here these are the values that individual clade is having. Okay, so this is all are the things clear to everyone?
Yes, sir.
So now any question? Dr. Sena, am I?
Hello.
Yes.
Sir, I have one question.
Yes. Yes.
So, the tree which is drawn by you is the basis of similarity decide that means similarity index will decide whether we should draw the tree or not, or because what I know that it is drawn for the new species.
No, that is that is not always. If you suppose if you have found Agaricus bisporus, okay,
Yeah.
so no problem. You have collected that thing from your locality. Suppose I have also found that Agaricus bisporus and have deposited the sequences in the gene banks, gene base, and I have collected the specimen from God. So as or anyone else have deposited that same species from any other place. So if there is any authenticated person who particularly deals with the taxonomy, and if he deposit any specimen in the GenBanks database, so if you prepare a phylogenetic tree with your sequence and the species that is deposited from other places, the same species sequence deposited from the other places and has been done by the taxonomists. So in that case, what will happen? Your sequence will cluster in the tree together with that specimen. Okay. If both the species are same, so you can in your paper you can discuss that that gives much more authenticity of your specimen. Is it? So that also could be the purpose of preparing the phylogenetic tree.
Okay, sir. Okay, thank you, sir. It was very nice presentation. I think we should get the recording so that we can do the practice whatever you have guided us.
Thank you very much.
Yeah.
Is there any other question from anyone else? You can ask, no problem.
Yes, sir. I have a question.
Yes, ask.
Sir, you have, while setting the parameters for the preparation of tree, you have said that bootstrap nearly 100 or 1000 or something, some value you have entered.
Yes.
What does it exactly refers to?
See, if we see the tree, can you see it?
Yes, sir.
Okay, so here if you see the support value, 83. What does this indicate? This indicates that these two species will cluster together 83 times if you prepare the tree for 100 times. That means out of 100 times, 83 times these two species will cluster together. And for the remaining 17 times, there might be alteration of these two species. Okay. So here, this value indicates that as it is 83, so this is supported. That means more than 50. In RAXML or ML tree, what we usually prefer, more than 90% support value indicates that the clade is a strong clade or strongly supported clade. Okay. Below that of 90, but above that of the 83, that is the moderately supported clade. That means it is moderately, this cluster of these two species is moderately supported. And below that of 80% to up to 70% is weakly supported clade. Okay. And below that of the 70%, usually scientists say that are unsupported clade. Yeah. So these are the statistical values that will denote how supported clade you have found with your taxon. Okay. So if suppose for an example, if you see, suppose for 7, that means out of 100 times, only 7 times these Volvarella, this species, Rava, clusters with this cluster. So see, it's a weak support. That means out of, if you prepare the tree 100 times, only 7 times it will come with this cluster. So here we cannot say that this species is the sister species of this one. Or if we write down in our manuscript that Volvarella rava appears sister to this cluster, but this cluster is weakly supported. Okay, but this position is weakly supported. We will have to say, as because the statistical support value is low, or the bootstrap support value is low for this cluster. Am I clear?
Yes, sir. So one thing, if we put the value 1000 in that box, then the same proportion we will have to check.
Yes, obviously. If you that depends upon you how much time you want to do that tree, and ultimately the bootstrap value will be so out of 100. Yeah.
Okay, but how we will decide the bootstrap value? What should be the proper bootstrap value that we should enter?
For ML3, 500 or more than 1000 is fine. Actually, we use 1000 times. Okay, you can also use 500, but not. I will not suggest below that of 500.
Okay, sir. Thank you. Thank you so much for such an elaborate explanation in focusing on each and every minute details. Thank you so much, sir.
Thank you. Any other questions from anybody else?
Sir, this is Sanjay here.
Okay. Fine.
Sir, my question is also related to the support value. And some support value is put on the node, and some support value are put in between the two taxa. Sir, can you please elaborate for me what is that value?
So here, this 7 indicates the support of this plate. Okay. See here, this 7 indicates the support of this plate, not here. This 5 indicates here. So you can design this. See here, the support value has come here. So if you want to visualize, or if you want to show the support value above the plate, so what you can do? Other than node level, you can manually do these things. You can add branch level, and in branch level you can select this PS. See here, all the values has been arisen above the plate. Okay, so these do not make the difference. The values will be same for that.
Okay.
Am I clear?
Yes, sir. Thank you so much. Thank you so much for very well presented.
Okay. Anybody else? Any other question?
Yes, sir. My question is that what is difference between bootstrap value and posterior probability?
No, posterior probability, you can get the posterior probability support value when you are using the Mr. base. Okay, and you can get the bootstrap support value whenever you are using POP or XML or Mega, these things. Okay, so usually that in case of posterior probability value, that is you will get maximum. It will be counted out of 1. So in case of posterior probability value, if you find out the value is 1, so it will indicate that 100% support. That is posterior probability PP1 equivalent to 100% of the bootstrap support value. Or suppose if you get PP value of 0.5, so it will be equal to that of the 50% of the bootstrap support value. Okay, so Mr. Base uses different algorithms than that of the your POP or Mega. Okay, so usually for posterior probability value, we usually say that more than a 0.9 is considered to be a supported plate. Okay, and below that of the 0.9, because Mr. Base uses such of the algorithm which usually shows much of the support value. So for an example, if you create a same tree with XML and POP, so what will happen for a clade? If you in XML ML3, if you find out that the support value is, suppose for example 70, but in Mega in Mr. Base you can get 1 here posterior probability value. So obviously this 1 is not equivalent to that of the 70%, isn't? That's why we often use 2 support values in the phylogenetic tree to show whether what are the values that are generating different softwares using different algorithms. Am I clear?
Okay, sir. Yes, sir. Thank you very much, sir.
Any other question?
Yes, sir. So in the phylogenetic reconstructions, how to highlight our samples that our particular fungi to be bold or to be in a different color like that?
What please say?
So how in a phylogenetic tree, how to highlight our fungi, our sample
How to highlight your
in a
sample?
bold?
What?
In a bold that our particular fungi which are included in the tree.
Okay, fine. So in Mega, you can get this option, edit taxa, or if you want to edit these things from the PDF file, what you have to do, you will have to convert this PDF file to the PowerPoint format. Okay. So as I have a software that is compatible to this PDF file has been installed. So I am opening that file, ML3. I am opening this file with a PDF editor. I will open with then Adobe Acrobat. So I have this one. Now I am saving it. Save as. So it will save it as a .pdf extension. So once this PDF extension has been saved, you can see this PDF sign here. So now I am opening it with Nitro Pro. You can do this task in online also. You will have to do what you will have to do. You will have to click convert PDF to PPT format. Okay, you can use this option. So as I have a software here installed in my computer, so I will not go in online. So I will manually convert my file here. So in here in Nitro Pro, you can highlight or you can convert it to the other format. How will I highlight? I am highlighting edit, then edit. See here, I am selecting this text. Suppose I want to highlight it. I have selected it. Control. I can use here ALMT Arial rounded. See here, I have highlighted my text. Okay. Is it clear now?
Yes, sir. Thank you.
So there are lots of available editing, softwares with the help of which you can, do this stuff.
Sir, does the branch length signify any genetic change or just we are manipulating it?
Yes. Obviously, that branch length signifies, the genetic distance between, both two species. See here, this is the, scale. That means this indicates 0.3 nucleotide distances. Okay. So here between these two species, although these Volvopluteus asiaticus and Volvopluteus michiganensis cluster in a same clade with 99% bootstrap support, but see here the branch length are varies from one another. That means the number of nucleotide changes. So this is the number of nucleotide changes, and this is the number of nucleotide changes for the Volvopluteus. So obviously, if we observe over the branch length, we easily say that there are nucleotide differences, and obviously these two might not be the same species. If both are the same species, we will not get such kind of less or broad long branch. Okay. So what will happen? See here, in case of Volvarilla diplasia, Volvaria volvacia, and Volvarilla volvacia, this Volvarilla volvacia has been deposited from China, this one from Thailand, and this one from India. All of them belongs to the same specimen. From India, whoever have done this thing is a wrongly identified. See here, it's a wrongly identified specimen. Okay, that's why this thing has been clustered with Volvarilla volvacia. So anyway, as all of them belongs to a same species, and see here, there is no branch length difference. There is a straight line, and with 99% clusters, all of your specimen have been clustered. Am I clear?
Clear, sir. Thank you very much, sir.
Hello.
Yes. Then.
Sir, my name is Ashwarya. I have one query. I have one indigenous culture that is somehow resemble to the one of the ATCC culture of the strain E. coli. And when I got the FASTA file, and when I aligned with the NCBI database, the first 10 sequences of the organism is not matching with the same ATCC number. Although the biochemical characteristics for that indigenous culture is showing the similarity with the ATCC strain, but the first 10 organism is not aligned with that ATCC number. What
No,
was
you
the
will have
conclusion?
you will have to see what is the percentage of identity with your specimen and the deposited sequence in the chain bank database.
Okay, so when the alignment result was came, that was just showing the similarity 99% with some of the E. coli, other strain of the E. coli, but not with that of ATCC strain that we are looking for.
That denotes that there is a genetic variation with your specimen with that of the deposited one. Obviously, there is genetic variation. That's why it's showing the differences. Or another thing you can do, you can manually check the quality of your sequences. If you see that, if you go through the everyone file, if you see the quality of your sequence is well enough, and again it is not showing the similarity, so you can see what are the bases, what are the differences, in which base do your sequence differs from the deposited strain in the NCBI database.
Okay. So is there any way to just or a specific primer in order to use specific primer to check the alignment? It's showing the 97% similarity, but the other organism that's showing 99% similarity, that was the closely resemblance match with that of
99%. Obviously, 99% similarity. The thing that is showing, that is much more similar to the US specimen than that of the that one. Might be there is some misidentification, or you might be misidentify your specimen.
Okay, sir. Thank you very much, sir.
Ladies and gentlemen, any more questions? No? Thank you, Dr. Arun. I think I would like to thank you very much.
Thank you very much, Dr. Senai, as well as the founder of the MycoAsia, for giving me such a excellent platform to connect to the micro lovers around the I would say around the world. Most probably. Thank you to all the listeners as well for for their special listening. I hope everyone have enjoyed the seminar. Oh, thank you.
Dr. Arun, you have been kind enough to spend almost 4.5 hours with us. Explain each details in a very you put a lots of efforts to explain the things to the audience today. I mean, I really appreciate your dedication towards the audience, and thank you very much for the hard work you have done. To prepare the slides, to explain the things. Maybe you might have skipped your lunch also. I'm not sure whether you even had your lunch.
I'll have it.
You will have it. Yeah, you will.
Yeah,
thank you. Thank you so much, Arun. And we will see you around. And you can please leave now. Okay, thank you.
Okay, thank you.
Ladies and gentlemen, are you ready for the quiz now?
Yes, sir.
Yes, sir.
Okay, let me give some information about the assessment quiz. You will have to answer 20 questions in 15 minutes. Okay, I will share the link in the chat box. Don't say I have not received the link afterwards. I'm going to share the link in the chat box. Have a look at the chat box right now. Here you go. You have 15 minutes. I will close the quiz in by 3:27. Okay, India Standard Time. Thank you. If people who do not want the certificate, they can skip this. They can go home. Thank you very much for your kind participation.